champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to start off with the next topic, that is nucleosome and the experiments for search of DNA. So previously we got to know that okay, the DNA might be the genetic material and it has a proper structure, and the structure and everything was identified, and also we spoke about the discoverers who were behind the finding of DNA. Okay, that was done, but now it's time to prove that DNA is the genetic material and DNA is being passed on from me to my offspring. Okay, so from one organism to another organism, or basically their offspring, it is transferred. It was proved by the experiments that we are going to see today. And also there was a confusion that DNA is pretty long. Then how can it be inside such small thing, that is the nucleus? Nucleus is like 10 to the power minus 9 meters. That is very, very, very short. So how can you expect something very long to fit in that particular space? So you will get answers to all your questions here itself. Okay. Now, the number of questions which was asked the previous year was 6, but if you see it was 10 and 9 in the previous years. So we can expect more number of questions as well, but in the previous year it was the highest. Okay, so this and few other chapters were on the top priority list because of the number of questions. Now this time we can expect more as well, right? Now this was the homework that I gave you. I hope my little champions did it, and let's see who all got it correct. Okay, so over here they have given this thing. So the figure gives an important concept in genetic implication of DNA. Fill in the blank, that is A, B and C. Well, this thing that you see over here, it is known as central dogma, about which we studied previously, and central dogma was put down by someone whose name was Francis Crick, who was also behind finding that DNA has a double-stranded structure. Okay, so James Watson and Francis Crick came together for giving the double helix model, and apart from that, I'm telling you, the Francis Crick was the one who gave the central dogma, which according to which the DNA is basically transcribed into mRNA, and an mRNA is translated to something that is known as protein. So proposed by is A. So please do not be confused. Over here A is at the end. Okay, so A has to be what, children? Francis Crick. Okay, and over here if you see A, it has to be Francis Crick, and B, obviously it will be translation. When you are converting RNA, not converting, basically translating the information in the RNA to form the protein, you call it as translation. And C is known as transcription, where the dRNA is helping in the making of mRNA. So D option is the correct option. I hope you got it, about which we covered in the previous session. So if you want to have a quick go through, you can go watch the video. Now let's start today's topic. So the length of DNA, what do you think is the length of DNA? If you remember, we studied that this is one loop of DNA, one turn of DNA. It can continue to the next one as well, but this will be the next turn. So this whole thing is one turn of DNA, and they have nucleotide base pairs, and this base pairs has a distance of, children, 0.34 nanometers. Okay, now if we know the number of base pairs, okay, if we know the number of base pairs and then we multiply it with the space in between them, can we get to know the whole length of the DNA? Yes, the space between them and the number of base pairs, if we calculate them together, if we multiply them, we will get to know the whole length of the DNA. So this is the thing that was exactly done. So taken the distance between two consecutive base pairs as 0.34 nm, or in meters if you convert, it will be what? 0.34 into 10 to the power minus 9 meters. It has to be lesser than meters, so that is why it is minus 9, please remember it. Okay. If the length of the DNA double helix in a typical mammalian cell is calculated by multiplying this. Okay, so let's calculate. So the number of base pairs which is there, over here I am filling this information as 6.6 into 10 to the power 9 base pairs. Okay, 6.6 into 10 to the power 9 base pair. Obviously remember, it cannot be minus 9. The number of base pairs will be way too much, so it will be 9 itself. Okay, so 6.6 into 10 to the power 9 base pair into the distance between them in meters, so that is 10 to the power minus 9 meters, you will get the result as 2.2 meters. This is the length of DNA. This is what, children? The length of DNA. But you know the twist? The twist is that if you talk about a cell and if you talk about the nucleus, this particular length of the nucleus is just 10 to the power minus 6 meters. Then now what? How can we fit this long 2.2 meters? 2.2 meters, can you imagine how long it is, right? How can you put this 2.2 meters of DNA into a very small space that is just 10 to the power minus 6 meters? For that, obviously, when you want to fit something in, what do? You coil them. Okay, you coil them. Similarly, our DNA is also made to get condensed, and for that we need the help of certain proteins. So you can imagine like if we have a rope and if you want to put the rope inside somewhere, we tie it up somewhere at regions, we tie it up so that it does not uncoil itself, it stays in the same format. Similarly over here also, instead of the tying thing, what do we have? We have certain proteins which keeps them in shape. Okay, so with this particular slide, you got an information that the whole length of DNA is 2.2 meters. And how did we get it? We calculated the number of base pair. It was 6.6 into 10 to the power 9 base pair, and the distance between them, that is 0.34 into 10 to the power minus 9 meters. Okay, it is in meters. This is in nanometers. Cool. Now let's see what happens. So I'm leaving this paragraph as of now. Okay, we'll come to that. First, let's talk about eukaryotes. Eukaryotes are the one which has the nucleus and everything, okay? Prokaryotes are the one which do not have nucleus. It has a specific region where the DNA is there. That is something different. Over here, we have a nucleus. So let's see how we can fit things inside the nucleus. So first thing that you need to know, children, is that there is a group of proteins. We call them as histone proteins. What do you call them, children? Histone proteins. Now, histone proteins is basically in the format of 1, 2, 3, 4, and in the back also they have 1, 2, 3 and 4. So basically, if I erase this thing, okay, I'll do one thing. 1, 2, 3 and 4. Over here also I have 1 and 2. Over here also I have 1 and 2. So basically, total if we see, it's an octamer. What do you mean by octamer? Basically, there are 8 sets of histone proteins. Okay, and on this histone proteins, our DNA comes and coils up. So it takes one round and then it comes back again. It makes the second round over here. So our DNA is basically coiled around something that is known as histone. Ma'am, what's the use of doing all this thing? Because you only think, if it was not coiled, probably it would have taken much more space like this. Now if it is coiled on something else, it's just rotating around something else, so it is utilizing much less space. So first thing that we have is histone octamer. Okay, that is the first thing that we have. Oh my god, where is it? Yeah, just a second guys, some technical glitches due to my hair, which is, yeah, fine. Now there is few more things that you need to know. That is, just imagine that this is the octamer. I'm not making the whole 8 ones, okay, and this is the next one, and you know that the DNA is coiling around it. Okay, similarly here also it goes and then coils around it. Fine. So basically what happens, you know that histone proteins are over here, but I want to tell you one more thing. There is one more histone protein over here as well in between them, and you call them H1 histone, or H basically stands for histone only, so I will write H1 protein. Okay, it is what? H1 protein. Will you be able to remember it? We have histone over here. This is also histone. It is made up of 8 type of different histones. Okay, different names are there. We don't need to learn that as of now. So it is known as octamer of histone or histone octamer, around which the DNA is wrapped. Now in between both of them, you have H1 protein as well. Now depending on the coiling, just imagine if it's like this. if it's like this, that means the DNA is in the supercoiled state. There is no space in between them. It's in the supercoiled state, okay? Whereas over here, it is not supercoiled. So I call this as very condensed form. This is very condensed form, whereas this is little condensed. It's not still condensed very much, okay? Now if you see, if I keep so many octamers in between, I find a structure like this, and this structure is known as bead on string structure. Bead on string structure, where you have the histone and you also have the DNA. Okay, and this bead on string structure have a scientific name to it as well. You call this whole structure as chromatin, my children. What do you call it? Chromatin. Okay, so this bead on string structure, where you have your this thing, what do you call, histone, as well as you have your DNA wrapped around it. Okay, you call this whole structure as chromatin. Now I want to tell you one more thing. Okay, what is this thing known as, where you have one histone octamer on this, the DNA is wrapped? You call just this one as nucleosome. This one structure is what? Nucleosome. And if you're asked this question, that how many base pairs are there wrapped around in one nucleosome, the whole structure, you have 200 base pairs, children. So the DNA over here will be having 200 base pairs. Please remember that. And this one structure, where you have the histone and the DNA wrapped around it, you call it as a nucleosome. So this is one nucleosome, this is another nucleosome, and in each nucleosome, how many number of base pairs do we have? 200 base pairs we have. Okay, and now if we see a long picture where you have so many nucleosomes arranged, okay, or basically when you have so many histones and the DNA wrapped around it, you call this as bead on string structure, which is nothing but the chromatin. Okay, now you might be confused, like, if this is chromatin, what is chromosome? So basically what happens, this is a loosened form. Now if I want to fit things in, what do? This bead sort of structure is arranged in more compact form. It is arranged in more compact form, and this structure will be now known as what? A chromosome. So is chromosome more condensed or chromatin more condensed? Chromatin is when, okay, the DNA is not plain, it has little amount of condensation, but still, if you look for chromosomes, it is a most condensed version of DNA that you can find, children. Okay, so this is about eukaryotes. So eukaryotes have octamer histones, histone protein, and the DNA is wrapping around histone proteins, right? And the structure that is formed is known as bead on string structure, which is also known as chromatin. Now I want to tell you one more very important. Why do you think that DNA is wrapping around this thing? So DNA basically is negatively charged. Okay, DNA is negatively charged. Fine. And the histone proteins that you see, okay, they have high amount of positively charged amino acids. So it's a protein, and proteins are made up of amino acids. So I am saying the histone protein that you see over here has positively charged amino acids. Okay, for example, you have to remember the name. One is lysine and the other one is arginine. What is it, children? One is lysine and the other one is arginine. It has the most numbers in the histone octamer, okay, or the histone protein. So lysine and arginine basically are positively charged amino acid, and when there is something negative, it comes and wraps around it, okay. Opposites basically attract, okay, opposites basically attract, and similar things repel. So over here, this is negative and this is positive. Fine. Now I think we can have a quick read of it. So yes, in eukaryotes, this organization is much more complex. There is a set of positively charged basic, okay, DNA, I'm writing here, DNA is acidic. Remember Friedrich Mischer, who said that there is an acidic structure inside the nucleus and he called it nuclein? Well, DNA is acidic and DNA is negatively charged. Whereas when you talk about histone proteins, it's the opposite of this. They are basic and they are positively charged. Now, the protein acquires charge depending upon the abundance of amino acid residues with charged side chains. So, it's not like all the proteins are positive, no, but they are positive. Why? Because of the charged side chain. So, histones are rich in basic amino acid residues such as lysine and arginine. So, they are basic, okay. Both the amino acid residues carry positive charges in their side chains. Now, in the structure, they are carrying the positive charges. You can remember like that. Now, histones are organized to form a unit of 8 molecules. 8 molecules, and what do you call it? Histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure that is known as a nucleosome. A typical nucleosome contains how many base pairs, children? 200 base pairs of DNA helix. Okay. Nucleosomes constitute the repeating unit of structure in a nucleus called chromatin. So, there will be so many nucleosomes coming together, and this structure is known as what, children? Chromatin, which is thread-like stained bodies seen in the nucleus. So, it can be stained, okay, it can be stained with basic dyes. This is acidic, other things can be, this can be used, stained with dyes and can be very much visible. Now, the nucleosomes in chromatin are seen as beads on string structure when viewed under the electron microscope. Now, if you are asked what are the bead shaped structure, it is nothing but histone, or basically you can say it's a nucleosome. Yeah, the whole thing where your DNA is also wrapped around the histone, right? So, you call it nucleosome. Now, what about prokaryotes? The ones that do not have, so we have a specific area where our DNA is present, and that is the nucleus. But what about them? That is the prokaryotes. So, prokaryotes you might have studied in 9th, okay. So, in them they do not have membrane bound stuff, as you know, okay. So, they do not have nucleus as well, because nucleus is membrane bound. So, instead of that, they have a region where the DNA is located. But what does it take for the DNA to be located there itself, and what do you call this region? You call this region as nucleoid region. Okay, what do you call this? You call this as nucleoid, and here also, children, there are positive charges. It is richly supplied in what? Positive proteins, positively charged proteins, okay. So, positively charged proteins, which makes them stay at one particular place. Fine. Now, over here. So, in prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell, DNA being negatively charged is held with some proteins that have positive charges in a region termed as what? Nucleoid. I told you, so it is there with the help of certain, held with the help of certain proteins which are positively charged. So, DNA in the nucleoid is organized in large loops held by the protein. Same thing that we discussed. So, I hope this is also clear to you. Yeah. Now, there is something else which is known as, okay, so this is something that I made. So, histone octamer, it is rich in lysine and arginine. Nucleosome contains 200 base pairs and it is beads on ring structure, and this is known as chromatin. Fine. Now, euchromatin and heterochromatin, what do you mean by that? So, basically, okay. So, basically, when you do not need the chromosome to be made always, they are sometimes loosely packed. Whenever we have work, at that point we get stressed, right? Until then we are like chill mode, right? Similarly, over here also, the chromatin will be like, let's just loosely be just there, okay? Let's not condense ourselves unless and until there is a need. So, the need comes when cell division arises. When cell division arises, obviously you cannot stay loosely spaced, okay, you have to be condensed because there is a lot of activities going on inside the nucleus. So, at this point of time, the chromatin which is there, which looks like this, will be now forming chromosome, which is the condensed form, okay. This has to be made. And when does it happen? Let's read it once. So, basically, the beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosome. So, you know cell division happens and there is M phase or mitotic phase, meiotic phase, where there is some phase which is known as metaphase. Okay, in metaphase, all the chromosomes, they arrange in a line. You might have already studied in grade 11th. Okay, if you have not, let me tell you. So, basically, the chromosomes have to arrange themselves in one line, and then One is pulled this side, other is pulled this side. So this is how the pairs they segregate. Okay. So over here, in the case of metaphase, the chromosome has to be made. So the packaging of chromatin at higher levels require additional set of proteins that collectively are referred to as non-histone chromosomal proteins. So at this point of time, when you want to condense it in more better way, you need the help of certain other proteins which are already there over here, onto which this our DNA is getting looping around. Okay, onto which the DNA basically loops around. So you can think like this. Something is there, okay, and this something is known as non-histone. This something is known as what, children? Non-histone proteins. And onto the non-histone proteins, the DNA which is there, it wraps around in a very much condensed format, making it a chromosome. Okay, so will you remember non-histone chromosomal proteins? Okay, NHC. NHC, very much important. Okay, so in a typical nucleus, some region of the chromatin are loosely packed. Okay, so if you see over here in this picture, you see the space between them is quite high and they are loosely packed. Okay, but over here, if you see, they are very much densely packed. So these guys, they do not take too much strain. So I say that it is basically, what do you say? Yeah, stains, this area stains slightly. This area stains slightly, where they show dark staining. So this area stains slightly, whereas they show dark staining. You call this area as Euchromatin, okay, and you call this area as Heterochromatin. Okay, so the densely packed, so I am writing, this is dark stained and they are densely packed region. Okay, now over here they stain lightly, they are Euchromatin and they are loosely packed. Now what is the use of it being loosely packed? When do you think that loosely packed the thing is necessary? Why do we have two types of things? So basically I said that DNA, you remember central dogma, where DNA is basically going to get transcribed into RNA and the RNA is going to get translated into the protein. So DNA to RNA, if have to be made, then there will be certain enzymes that has to be coming over here and transcribing the information which is stored in the DNA. Now if you do not give them space, okay, if you do not uncoil the DNA, then how can they work on the DNA? They have to basically sit on the DNA, they have to basically sit on the DNA and read the whole thing, making the DNA help in the formation of something that is known as mRNA. So I say that this particular region, the loosely packed region, they are transcriptionally active because here transcription is taking place. So what can I write, children? They are transcriptionally active. Transcriptionally active. Whereas here, if you see, no transcription takes place. They are loosely packed, they do not give space. They are loosely packed, they do not give space. Will you be able to remember it? Euchromatin and heterochromatin. Cool, let's read it. So, in a typical nucleus, some regions of the chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are known as heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. So, I can say no transcription, it is inactive. Easy, no? It's not very difficult. It's easy. If you listen to me very carefully with a zeal that, okay ma'am, we will try our best to understand things, everything is possible in your life. So have that zeal in yourself, have that good amount of motivation in yourself, have that belief in yourself that you, if you decide, you can do anything. Yeah? So that is all about euchromatin and heterochromatin, and that is all about the structure of nucleosome as well. Done? Now going ahead, we are going to do an experiment. It's a very, very nice experiment. It is known as transforming principle experiment. It was given by none other than Frederick Griffith. Okay, so with love, we also call it Griffith experiment. What do you call it? Griffith. I need to check the spelling because I mess up a lot of times. Okay. So it is Griffith's experiment. His name is Frederick Griffith, and this was given in 1928, children. Please remember the years. Okay. Now, according to him, he did an experiment with Streptococcus pneumoniae, the bacterium responsible for causing pneumonia. We studied in human health and diseases, right? So, we have Streptococcus pneumoniae and we are going to work on that. So, he witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism, bacteria, had changed in physical form. When Streptococcus pneumoniae, that is pneumococcus, you also call it pneumococcus, okay? Bacteria are grown on a culture plate. Some produce smooth, shiny colonies, while some other produces rough colonies. So, I will tell you the basic difference between them. So, basically, just imagine there is a bacteria, and this bacteria, few of them will be like, we cannot work in this situation, we have to be very glossy, very shiny and very stuff like that. So, basically they have a very shiny coating outside them, okay. This is shiny coating that is outside and it is very smooth, okay, it is very smooth and very shiny. So, we say no, the things that looks good does not always have to be good. So, you should not be judging anyone by their appearance, right? So, it's shiny and smooth. I'm telling you why it's not that good later on. And then you have something else, which is kind of, they are rough, okay, and not shiny obviously. So, this will be rough and not shiny. That is something else, okay. This smooth shiny thing that I am talking about is what is the mucous membrane. It is made up of a polysaccharide. Polysaccharide as in carbohydrate. So, it is made up of a polysaccharide. And now, when they divide, obviously there will be so many of them together. So, I am just making something like this with a green color coating outside. Okay, so that becomes what? A colony of this one, that is smooth shiny ones, and the other one are basically colony of rough ones. What happens? This is because the S strain bacteria. So, this is a strain of bacteria, this is another strain of bacteria, but of the same bacteria, that is pneumococcus. So, pneumococcus has two different strains. One is S strain, okay, one is S strain and the other one is R strain. S stands for shiny smooth and R stands for rough, okay. Now, this is because S strain bacteria have a mucus coat while R strain does not. I already told you. Mice infected with S strain virulent, okay. So, this strain, even though it's shiny and smooth, it is the one that causes disease, that is pneumonia. So, I say that this is virulent. This is what? Virulent, whereas this is non-virulent variety. This is non-virulent variety. Okay. So, mice infected with S strain, that is virulent form, die from pneumonia infection, but mice infected with R strain do not develop pneumonia. Okay. Griffith was able to kill bacteria by heating them. Griffith thought, hey, this is the guy, I need to kill it. So, he tried to kill it also by heating them. Okay. By heating, what happens? Basically, the DNA and everything which is in the form of this, okay, DNA which is in the form of this will be uncoiling itself and will be two separate strands. Okay, now if DNA is not there, obviously the proteins won't be made. If proteins are not made, okay, the DNA is not proper and the proteins are not made, then obviously can an organism survive? No, even we cannot survive. Okay, so he tried to heat and kill them. Now, okay, he observed that heat killed S strain bacteria injected into mice did not kill them. So, when he killed this strain and tried to put it in the mice basically lived, it did not die. I will tell you everything. When he injected a mixture of heat killed S and live R bacteria, the mice that died. What happened? Basically, he took heat killed S, but he also took the R strain. Okay, and then he put both of them into the mice and then he found that the mice died. But you have heat killed this one, then how did the mice die? Basically, probably this guy have transformed this guy into S and now it might have got virulent. Okay, that might be possibility, right? So, moreover, he recovered living S bacteria from the dead mice. He also saw that in the mice there was S, but he put the heat killed S, then how did S come? So, probably this R might have got transformed. Moreover, he concluded that R strain bacteria had somehow been transformed by the heat killed S strain bacteria. Some transforming principle he said there was some transforming principle, okay, that was transferred from this guy, that is heat killed S strain, to the R strain, which enabled the synthesis of the smooth polysaccharide coat and became virulent. So there was something that was transformed, this transforming principle, which basically helped in making a coat around the rough strain, making it smooth now. Okay. This must be due to transfer of the genetic material. However, the biochemical nature of genetic material was not defined by this experiment. So even though he gave a basic idea, a lot of things were still unclear. So let's see basically what he did. Over here, he took S strain. Okay, I'll start with R. He took R strain, and this was live R strain. He put it in the mice. The mice lived. Because R is what, children? R is non-virulent. R is non-virulent, so obviously it will survive. This was the first case. Okay. Second case, he took live S strain, okay, and he put it in the mice. The mice basically died. The mice died because this particular strain that you see over here, it is virulent. Okay. Now what he did is that, okay, he found that, okay, this could be a problem. So what do? I heat kill S strain bacteria, and then I put it in mice. Surprisingly, the mice lived. That means because of heating, some changes was happening over here, which is why it's not virulent anymore. So I'm rating non-virulent. Okay. When it went on, and Griffith was like, Frederick Griffith, I am not understanding anything. I should be doing something different. So what did he do? What did he do? He took the fourth step. That is, he took heat killed S strain, and he said, I also want to take R strain. But this is normal I strain, okay? He took live R strain, which was not causing any disease in the upper area, right? And he put it in mice, and he found that basically the mice was dead. How can that be possible? There is some problem. So he concluded with his experiment that probably the heat killed S strain is passing on transforming principle to R strain, which is why now R strain is getting converted to S strain. It is making a coat around it, okay, with the help of this converted thing, whatever is transformed. With the help of this, it is making a coat around it, and it's getting converted to S strain, and this S strain is the one that is actually put in the mice. Basically, this S strain is the one that makes the mice dead. Okay, so he concluded this thing. He said that this might be the genetic material, but he did not have anything else to say. So this was done by Frederick Griffith, and this is known as transforming principle experiment. Obviously, people might have worked on it. They wanted to find out what is that thing that is getting passed on. We still did not know whether it's DNA, RNA, protein, whatever. Okay, so comes the next set of people who went for the biochemical characteristics of transforming principle. They want to know what is the chemical feature, what is the structure of this thing, not the structure, basically chemical feature of this thing, which is known as transforming principle, which is getting transferred. So the next set of experiment was done by Avery, MacLeod and McCarty in the year 1933 and went up to 44. So 10 years. It's not a one-day thing. For 10 years it worked on like that. So Avery, MacLeod and McCarty, all of them, they worked on this thing. So now let's see. So what happened? Let's see that. So prior to the work of Oswald Avery, Colin MacLeod and Macklin McCarty, please remember Oswald Avery, Colin MacLeod and Macklin McCarty. Before that, I'm so sorry, I just want to write pneumococcus here because I think you will forget. So it is pneumococcus on which this particular experiment was done for doing the transforming principle stuff. Okay. Now over here, Oswald Avery, Colin MacLeod and Macklin McCarty in the year 1933 to 1994 worked on this, and the genetic material was thought to be a protein. So prior to this, prior to their work, it was thought the genetic material would have been a protein. But they worked to determine the biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals like proteins, DNA, RNA, etc. from the heat-killed S cells to see which one transformed live R into S cells. So basically what did they do? They purified, as in they took, for example, this was heat killed S strain, okay, and they took the DNA out of it, they excluded the RNA out of it, they took the protein out of it, okay, to check which one was actually causing the conversion. So they put this into R strain, they put this into R strain, they put this into R strain, okay. They discovered that DNA alone from S bacteria caused the R bacteria to become transformed. So they saw that when they put DNA, that's when the R bacteria, which was like this, it formed a layer around it, and now it has become what? S strain. Whereas they did not have any change, because you are putting DNA and you are having protein. Okay, so this was given as first thing by Avery, MacLeod and McCarty, but this was not enough. Okay, this was not enough. Probably there could be something else also which was getting transferred along with the DNA. So you never know. So please remember, it was taken from the heat killed ones. Okay, now they discovered that DNA alone from, okay, they also discovered that protein digesting enzymes, proteases and RNAs, that is RNA digesting enzyme, did not affect transformation. So now the next thing that they did was, okay, that was the first thing that they did. Second thing that they did was, so over here it is the S strain, the heat killed one, okay, and this is the R strain. Similarly S, similarly again R, S, R, all of them they are heat killed. Now they treated this strain with protease, okay. Protease are protein digesting enzymes. Protein digesting enzymes. So basically whatever proteins are there, that would have been digested. So none of the proteins would be going ahead, right? So this conversion was anyway not taking place by doing this thing. Okay. Next thing. Okay, just a second. Let me think. Okay, wait. One thing. Okay. Basically, small change. Okay. So when this was done, still it was making the coat. So this one was changing into S strain. This one was changing into S strain, that is the virulent one. Now next thing that was used was RNAs. So when RNAs are used, it is what? RNA digesting enzymes. RNA digesting enzymes. At this point of time also, there was no change. It was still what? Transforming into virulent form. Okay. The next thing that they did was, they took DNAs, which is what? DNA digesting enzymes. They took DNAs, which is DNA digesting enzymes, and what they found was kind of extraordinary. That is, now the R strain did not get transformed into S strain. That means it's because of this that it was getting transformed into S strain. So when DNAs was used, all the DNA got damaged here. So obviously there is nothing to get passed on from here to here. So this was not getting transformed. Whereas over here, when we were using protease, when we were using RNAs, everything was getting transferred. There was no effect that was seen. Everything was all good over there. Okay. So that is the thing. This was done by Avery, MacLeod and McCarty. Okay. So that is exactly written over here. They also discovered that protein digesting enzymes and RNAs did not affect transformation. So the transforming substance was not a protein or an RNA. So prior to that, it was thought that it was a protein, but now you got to understand it's not a protein, it's not an RNA. So digestion with DNAs did inhibit transformation, suggesting that DNA caused the transformation. They concluded that DNA is the hereditary material. But please remember, not all biologists were convinced. Even at this point also, after Avery and MacLeod and everyone did this, still the scientists were not happy with this. They were not convinced. So we need a final proof, and that is a question also, that is a PYQ also. It always comes that who are the people who are behind the final proof that DNA is the genetic material. DNA is the one that is getting transformed. So let's see who were those people. The genetic material is DNA was said by Hershey and Chase in the year 1952. So you see the years going around. It was starting in 1928 by Frederick Griffith, and then from this period to this period, that is 10 years, they all worked. Now in the year 1952, that is almost 9 years, 8 years, around that, they started working, that is Hershey and Chase. They said, they started working earlier, but they said this thing, the DNA is the genetic material at this point of time. So there's an important point that is given, the unequivocal proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase in the year 1952. This is very much important. Unequivocal is very much important. So unequivocal, there is no one to go and say that this is wrong. Okay, there was no one to say that. It's perfect way to say that DNA is the genetic material. So they worked with viruses that infected bacteria, and these were known as bacteria called bacteriophages. Okay, they worked with viruses that infected bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the virus that entered the bacteria. So basically I am saying that there is something which is a virus. Okay, it has legs like this, and this virus is known as a bacteriophage. Okay, what do you call it? You call it a bacteriophage. Please remember, bacteriophage is not the bacteria. Bacteriophage is going to affect a bacteria. And according to the information given as of now, whatever DNA is there or whatever genetic material is there, this was a DNA based virus. Okay, please remember, this is a virus, and virus usually can have DNA or RNA, but this was DNA based virus. Okay, so this DNA or genetic material, whatever it was there, when it affects the bacteria, it transforms it over here, it passes on onto here, and the bacteria thinks that okay, this is my own DNA, and keeps on replicating, making more copies of it, and lastly forming small bacteriophages. Because you got the DNA and everything, with the help of DNA you can make the protein. So proteins, okay, I'll tell one more thing. This thing that you see over here, it's made up of protein, and you call this as capsid. And viruses are like that. They have the genetic material and they have a protein coating around it, which is known as capsid. Not like us. We do not have cell membrane, stuff like that. Okay, they just have a capsid, that is protein. Apart from that, they can have other membranes. Some viruses have, some do not have. But this is the basic thing. Okay, they have a capsid. Inside the capsid, they have the genetic material. So the capsid is made up of protein, and inside that they have the genetic material, and they found that okay, this genetic material was passed down. Okay, and with the help of this DNA helps in the formation of proteins, right? So with the help of this DNA, proteins are made, and now you have small chunnu new bacteriophage inside this. That's how things work over here, and then they are released. Okay, so Hershey and Chase worked to discover whether it was protein or DNA from the virus. Okay, they didn't know that whether this or this was getting passed down. Okay, so they grew some viruses on a medium that contained radioactive phosphorus and some others on a medium that contained radioactive sulfur. Okay, so sulfur, children, is a part of protein. Okay, in the structure of protein you will be finding sulfur there. Whereas when you talk about phosphorus, you know, we have studied on the fifth end, on the fifth carbon, we have phosphorus, which goes and attached to the third carbon of the next deoxyribose sugar. You remember that, right? In the formation of DNA. So we have phosphorus, so DNA is there. So phosphorus is there in the DNA. We got that also. Okay, so DNA has phosphorus, proteins have sulfur. Okay, and what did he do? He took two things. Okay, he said that I want to grow a bacteriophage, okay, a type of bacteriophage inside what, children? Inside a medium with radioactive sulfur. Okay. Now, when you treat with a medium that is having radioactive sulfur, what will happen, children? The radioactive sulfur, wait. Now, it is like this. It is radioactive, it has changed. Okay. Now, the capsule looks like this. Further, I have another bacteriophage and I grew it in a medium which had radioactive phosphorus, and phosphorus goes and affects the DNA. So, if the normal DNA was like this, okay, so the radioactive form of DNA, I am making it in green itself. So, this is the one that has the radioactive DNA. So, over here we have the protein which was changed, and over here we had the DNA which is now changed. Okay. Now, it allows the first step, that is infection. It goes and infects over here. This will go and infect a bacteria. Similarly, this will also go and infect a bacteria. That is the first step, that is known as infection. Now, after infection, what do they do, children? After infection, what do they do? So, basically, this guy came over here, it sat on this. Similarly, this also sat on this. It passed on the genetic information, that is whatever is there. For example, assuming it's this, okay. Over there also it passed down things. So, it's this over here, cool. And now, the next step was blending, where we want to separate. So, after sending things down, okay, there is nothing inside the bacteriophage, it's just there. Over here also the bacteriophage is just there. So, we want to separate this. So, that will be the next step, which is known as blending. In blending, what happens? Basically, whatever is stuck on the bacterium, it gets separated. So, the proteins, if you see, they are not passed down. Okay, the proteins, if you see, they are not passed down. While blending, it's all gone away. Okay, so while blending, everything goes away. Now, the next step is known as centrifugation. Okay, next step is known as centrifugation, where you spin things very fast. Okay, in such a way that the liquid portions get separated and the cells, they get separated. The liquid portion and everything, you call it as supernatant. Okay, liquid portion, you call it as supernatant. And here, in the case of this, the supernatant will be having the capsid proteins, because that was blended away, right? Oh, now over here, if you talk about supernatant, okay, it did not have the capsid, okay, it also had the capsid protein, but was this radioactive? This is radioactive. The supernatant is radioactive because the capsid protein was basically radioactive, right? This was radioactive, so that will be radioactive as well. On the other hand, this one was not radioactive. If you talk about the supernatant, it was not radioactive, since the protein was not the one that was radioactive over here. But the twist that was there was the cells, when it was, so supernatant is different, cells which will be dense, they will be coming down. So, cells, that is bacterial cells, it was coming down. Over here they found that it was not radioactive, whereas over here the cells, if you see, they were radioactive. Why? Because it's a DNA that has got passed down. So, obviously, they will be helping in formation of more bacteriophages inside. Okay. So, DNA which was stacked, it is getting radioactive, and DNA is stacked inside the cell. So, inside the cell, this DNA is there. Okay. If it was not passed down from here to here, the cells would not have been radioactive. But now the situation is different. It is radioactive. That means the DNA that is getting passed down from here to here. Did you get it? So no radioactivity detected in supernatant, but yes, radioactivity detected in the cells in the case of the P32 affected things. Okay, so radioactive P32 is basically the one that is being used over here, the radioactive version of phosphorus. Over here S35, okay, just a second, okay, I'll write it in black so that you can see it easily. So this is 32P that was used, whereas here it was 35S. Remember, S looks like 5, so you can remember like that. 35S and 32P was used over here. 35S goes and binds with the proteins, whereas over here it goes and binds with the DNA. Okay. And now if you see, the cells were radioactive, the supernatant wasn't. Over here you saw that the cells were not radioactive, but the supernatant was. So it's the opposite of things that is happening. And with this experiment, he came to a conclusion that, or they came to a conclusion that DNA is something that is getting passed on and not something else. Okay, so that is what I exactly wrote over here as well. So I'll just read through. So viruses grown in the presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, because DNA contains phosphorus, not protein. Okay, similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain sulfur. It's a protein that contains sulfur. Okay. Now, radioactive phages were allowed to attach to E. coli bacteria. So, yes, I forgot to tell that. What is the bacteria that was used over here? It was E. coli, children. Okay, it was E. coli bacteria which was used over here for the experiment. Now, then the infection is preceded. The viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. That is the third step. Okay, bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses. DNA is therefore the genetic material which passed on from the virus to the bacteria. Okay, did you understand this? So bacteria, a cell, same thing. Please understand that. If you say bacteria or you say the cell, so bacteria is a unicellular thing, so which is why you can also call it a cell at times. Be specified, it will be much better. You're talking about E. coli, which is the bacteria over here. Okay, so we saw there were 3 experiments being done. First was Griffith's experiment, where basically he used the S strain and the R strain of pneumococcus bacteria on the mice, okay, and he found that S strain was the violent one and something was getting transformed from the heat killed S strain to the R strain. Later, Avery went to prove that, okay, it's not the protein, it has to be something else. So they did their own experiment where they treated the heat killed S strain with RNAs, protease, and still it was getting transformed. The R strain was getting transformed to S strain. So it was not RNA or protein that had to do anything with it. But when they put DNAs, when they treated with DNAs, now things were kind of different. Okay, it was not getting transformed. But people were not happy with this experiment. Still, they didn't believe. There were so many biologists. Obviously, we do not believe on things very quickly, right? So then came Martha Chase, that is Martha Chase and Alfred Hershey in the year 1953, right? 1953 they came, right? 1952, okay, 1952 they came and they did their own experiment, okay, on bacteriophage and the E. coli. Bacteriophage is a virus which infects the bacteria. Please remember that. Okay, and they used 2 varieties of this thing, medium, radioactive medium. One had radioactive phosphorus, which is 32P. Okay, and one had radioactive sulfur, which is 35S. Okay, so the S, that is sulfur, goes and attaches to the proteins. Okay, it's a part of a protein. Okay, and now if you talk about phosphorus, phosphorus is a part of a DNA. So obviously it will be showing in the changes in the DNA. Later when he infected, he saw that the bacterial cells which was showing radioactivity was the one which were grown in a medium where the bacteriophage had this thing, that is P32. Okay, that means DNA was the thing that was getting passed on because phosphorus is there only in the DNA, not the protein. Cool? Works? Now let's solve certain questions. Yeah. So first question. The association of histone H1 with nucleosome indicates that. Tell me the answer now. I'm not solving over here, we'll solve it later on. Okay, tell me the answer. The association of histone 1 with the nucleosome indicates that DNA replication is occurring. DNA is condensed into a chromatin fiber, DNA double helix is exposed, or transcription is occurring. What? Okay, this is first answer. I hope you're answering me. I'll see it. Okay. Second, the unequivocal proof of DNA as a genetic material came from the studies on what? On what? Bacterium, bacterial virus, fungus, viroid, what? Second question. Okay. Please pause and try to do it by yourself. Third, the final proof for DNA as a genetic material came from the experiments of Hershey, Chase, Avery, MacLeod, McCarty, Hargobind Khurana, or Griffith. This is the third. Easy question, no? Yes. NEET 2017 plus 4 marks. Now, fourth question. 1 turn of helix in a B-form DNA is approximately how much nanometers? So this we studied in the previous lecture, but still, you can still answer because I told you about what is the distance between the base pairs. Okay, and now I am giving you a clue. If in one helix there are 10 base pairs, what is the length? And each base pair has a distance of 0.34 nm. Okay, nm. What is the distance or what is the length of the whole DNA? You have to tell me that. Cool? Going ahead. Which one of the following pairs of nitrogen bases of nucleic acid is wrongly matched with the category mentioned against it? This is again from the previous lecture. Which one of the following pairs of nitrogenous bases of nucleic acid is wrongly matched with the category mentioned against it? So you have two things that is, okay, I'll tell one help. A always matches with T and G always matches with C. Now you can tell me which is purine and which is pyrimidine. Whatever. Did I actually tell the answer in some other ways? These are the 5 questions. Please let me know the answer. Now I'm going to solve it. Okay, so the association of histone H1 with nucleosome indicates. So this is a nucleosome, this is a nucleosome, imagine, and this is a new DNA that is getting wrapping around. Okay, and you have the H1 histone over here. So if its association is there with the nucleosome, it's very close. That means the DNA is condensed into a chromatin fiber. If it was not there, then probably H1 won't be there. Okay, it is forming a chromatin fiber. This is a chromatin fiber, children, where you have everything there. Okay, DNA helix is exposed, no. Transcription is occurring, no. You are asking about association of H1 with the nucleosomes. That means they are close. So it has to be condensed to form a chromatin fiber. Cool? Now, unequivocal proof of DNA as genetic material was given proof of DNA as genetic material came from the studies on what? Bacterium, fungus, viroid, bacterial virus. It was bacterial virus and the name was bacteriophage. Done? Now, the final proof of DNA as genetic material came from the experiments of whom, children? Hershey and Chase. They chased behind the reality. You can remember like that. They were the people who chased behind the reality. They want to find out the answers anyhow. So it was Alfred Hershey and Martha Chase in the year 1952 who did the finding. Okay. 1 turn of helix in B form DNA is approximately, B form is the commonest form. Okay, and it's approximately, children, equal to 0.34 nm is the distance between each, and there are 10 in 1 turn, I said. So it will be 3.4 nm. If you're not understanding, I can draw it and show you. So over here I am saying 1 turn. This whole thing is 1 turn, okay, or 1 loop or 1 turn, and it has 10 pairs of bases. 10 base pairs, okay, 10 base pairs, and the distance between each base pair is 0.34 nm. So to calculate this, I multiply it, I got 3.4 nm. So this is the correct answer. Cool. Now, which one of the following pairs of nitrogen bases of nucleic acid is wrongly matched with the category mentioned against it? So, adenine and guanine are purines. Adenine, thymine, purines, we have to find wrongly matched. So, I'll tell you, one is purine, the other one is pyrimidine. So children who are confused as of now, I request you to please go watch the video if you have not watched it earlier, where I have discussed the detailed structure of DNA. If you are not seeing that, Baba, you won't be able to understand the chapter really well. So you need to know the basic things, right? So please have a quick view of it, okay? It's a really nice video. Now, purines come under, A and G, that is adenine and guanine comes under purines. Under pyrimidines, you have T and C, TC. Okay, TC for pyrimidines. You would be getting TC if you are joining pyrimidine group. Thing like that, whatever you want to say. So guanine and adenine purine, it is correct. Adenine and thymine purine. Adenine and thymine are the purine. So this is wrong. Okay. Now thymine and uracil, pyrimidine. So in the case of DNA, it's like this, but in the case of RNA, instead of T, you have U. So how many pyrimidines do we have in total? U, T and C are the pyrimidines that we have in total, including the one that is there in RNA as well. Okay. So thymine and uracil are pyrimidines. As of now, let me replace. So either it can be U or it can be T. Okay. Thymine, uracil, pyrimidine, uracil, cytosine, pyrimidines. That's all true. This one is wrong, children. 2008 question. Easy? Happy doing the questions? I feel that it's a very good thing that we are doing, right? Practicing questions, trying to find out the answers. So now one question will be there for you for doing it as a homework. That is, what are the structures called that give an appearance on beads on the string in the chromosome when viewed under the microscope? Genes, nucleotides, nucleosomes or base pairs? Okay, that is a question, homework question for you children, my dear little champions. Apart from that, okay, so we are thinking the shorts thing probably might not be there for some time. Okay, rather than that, we will be utilizing those shorts for something else, like solving questions and stuff like that. So I'll be giving you homework here itself. So this is the homework that I want to give you for the next lecture, which will be on the experiments of what? How can I forget? Yeah, we will be talking about the comparison between DNA and RNA, and also we will be talking about one more thing, that is basically the semi-conservative nature of DNA and how was it proved. So you can go have a thorough read for all those pages that is there in your NCERT. I'm telling you again, the comparison between DNA and RNA just after the Hershey and Chase experiment, you can see that. Okay, and it goes up to the next experiment, which was given by Michelson and Stahl. So they were the two people who gave an experiment, and on the basis of this you can try to solve these questions that you have given over here, and you can let me know the answers here itself. If you're not able to understand things, obviously I'll be solving in the next lecture as well. So you can take a screenshot. I'll just move away. Okay, I hope it works. Yeah, so that's all, children. I hope that this class was helpful for you. If you think the same, then do not forget to comment down and also to subscribe to our channel. I hope that you have a very blessed day. Have a good weekend as well. So bye bye, take care.